Afternoon, Suzuki Science. I am going to complete the membrane lab. I'm going to remind you where to find it. So I'm doing a primary screen share. You go to the calendar. You go to today's date because we're doing this on Thursday. Right, complete osmosis diffusion lab. Right. And then on here, this was the main lab that you guys did as a group today. This is the lab I started the demonstration. I'm going to complete the demonstration now. So I click on this link here, and it will open up my lab. Now, I generally don't like working in the doc viewer. I like working in the Word file itself. So personally, I would download the file. I have the file already open on my computer, so I'll go to that. So this is that membrane alcohol lab. You might recall that I used 3 different alcohols. I used methanol, 1 carbon, ethanol, 2 carbons, and propanol, which has 3 carbons. And we want to determine what is the effect of alcohol on biological membrane. So this is a great connect between the membrane science that we just talked about in class today and moving forward into homeostasis. Right? We always want to connect these biochemical fundamentals to some larger applications with the human body and other biological concepts. Okay? So, assuming that I am being recorded, let me jump down to the tool that I'm using. So you recall that I took my beets pieces, I cut them up, and I put them into the test tubes. I've been soaking them for a little while. Unfortunately, I didn't get a chance to mix them, which would have been a useful thing to do, but I was doing a lab. And just by looking at the 0 versus 100, and let me get a white sheet of paper here. Just by looking at the 0 versus 100, you can see that there's a qualitative difference between the two. Like, you should be able to see that. But we don't want to rely on qualitative data. We want to use a number. So we use a special colorimeter, the special probe, and it will give me the exact measurement, the exact absorbance. Now, because the beet is made of that anthocyanin, which is reddish, you haven't learned this about the chemistry yet. You see on the screen, I've left this little blurb for you, that generally when we're looking at absorbance, we look at the color wheel and we look on the other side of the color wheel. And we take the complementary color. That's what I'm going to measure. So I'm not going to explain the science, but I'm going to allow you to either read the blurb I have on there to get a bit of an explanation, or do your own research, or learn this in chemistry eventually. Okay? So since I want to determine how much red was released, I'm going to measure blue absorbance using the special colorimeter. So I'm going to connect through my SparkView. Make sure it's turned on. There's my colorimeter. The first thing you must always do with the colorimeter is you must, because these hold fingerprints very well, I always want to use a special Kim wipe to make sure it's clean. On the screen here, you can see my SparkView app here. So I want the blue absorbance. Green was kind of on the other side of that color wheel as well. So I guess I could do blue and green just in case. Right? I definitely don't want red. I don't want transmittance. I want absorbance. So blue should be fine. Let's just stick with blue, right? Because that's on the other side of the color wheel. Now, I care about the value. I don't care about the graph. So I'm going to click on the digits here. When you use the colorimeter, you must calibrate it first. And you want to use whatever base solution that you're using. So because I am using methanol, ethanol, and propanol, I really should use that as my base for this calibration. In this tube yesterday, I did put in some ethanol, so let me just clean this off. Right. So I'm going to calibrate it with the ethanol. If you look at the colorimeter, you'll see that there's a light. The light's coming from here, so I want the clear side towards that light. I'm showing this to you because we will be doing a plant lab eventually, and it'll be using a spec as well to measure the pigments. So, that in there, close, calibrate. So wireless, everything's there, insert blank with a solvent. Right, so I'm going to click calibrate. I got a check mark, I'm going to click done. So I'm done with this. Really, I should have one for methanol, one for ethanol, one for propanol. I just used ethanol. Okay. Now, I'm going to take the solutions from here, I'm going to put them in these cuvettes. So take it in, put my cuvette. I don't know how well you guys can see it, so let me just change my screen angle, my camera angle. Hopefully you see it now. What I'm going to do is I'm just going to do each one of these, as opposed to doing them and measuring. I'm going to do 1. So that's my 0. I'm going to do my 10%. I'm going to pour it in, it'll be easier. So that's my 10% methanol. Pour this in. My 20%. Putting the lids on in case I knock it over, it won't spill everywhere. 30%. Lid back on, and my 40%. Okay, so I've now poured off my methanol from 0 to 40%, and now I'm going to clean each one. Put the lid on. Clean each one. Okay, now I'm going to put it in my colorimeter. I'm going to hit start so I can actually get a value. So 0.127. So I'm going to record that on my chart that I have open somewhere. So methanol. I have 0, 10%, 20%, 30%, 40%. Nothing. So at 0, my blank, it read 0.127, I believe, 0.126. Okay, take that out. I'm done with this, so I'm going to put it over here so it's out of my way. I'm going to put in the next one, close it, measure, 0.100. Next one, 0.180. Now normally when we do this, there's an entire class, right? So if there's any kind of erroneous data point, typically it will average out. I'm doing this by myself, so if I have an erroneous point, I should be able to see and make note of that. Okay, and my last one, my 40%, 0.339. That's significantly more. 0.339. So when I cut my beads, my premise is that each of my little bead pieces that I cut, they're all exactly the same. But obviously, I'm not that accurate. I wasn't using a very fine piece of equipment. I was using a big giant butcher knife and then cutting them to the best of my ability. So there's going to be some discrepancy there as well. Okay. So now that I finished with those, I could see that my 0% alcohol had 0.127. My 10 seems like I had a little bit of an erroneous result there, so who knows what happened. But as we went up to 20, 30, and 40%, the 40% had much more damage than the 0%, as expected. Okay, and then just looking at these, hopefully I can see this on the camera. Just looking at these, you can see the white sheet of paper here. You can see that there is a difference from 0 to 40 just by looking at it qualitatively. Right, so I'm done with those. Now I'm going to do the same thing with the with the ethanol and the propanol. So this I won't record. You don't need to see me do this. I'm going to pause the thing right now, and then I'll come back on when I've done all three. All right, so I finished pouring these in, so I'm measuring them now. I thought I'd bring you in while I'm measuring them. So I did the first one, it was 0.168. So here's a 10% ethanol. And that is 0.217. Where's my graph? 0.217. And the 20% is 0.206. And the 30% is 0.416. And the last one, 40% ethanol, is 0.935. That's high. And once again, I can see that when I look at them on camera, just qualitatively, I can see that there's a difference from 0 up to 40%. So that's my ethanol. And already looking at my methanol and ethanol, I can see that there's a difference between the alcohols. So now I'm going to do the propanols, and I'm going to pause it momentarily while I dump those in. All right, I'm back at it. So I've done the propanol. I've poured it into the cuvettes. That's the proper name for these little test tubes. And I'm placing them into the apparatus. So the 0 is 0.156. So really, all these ones should be the same, right? Because they're all the same thing. They're all water with no alcohol inside of them. So you can see there's a little bit of discrepancy, and that discrepancy must be due to the way I cut the beet. That's 1. So this is now 10%. This is 0.204. Okay. And now we'll go with 20% propanol. And this one here is 1.254. That's a huge increase. Do the 30% now. 1.850. And now the 40%, if everything worked out well, this will be the highest absorbance. And we got 1.993, 1.998. 1.998. So that is the result. Those are the results, the observations, and the results for our membrane lab. So we can clearly see out of the 3 alcohols that there's a trend. This 0, that should be the same number because all of them are just water. So if there's any difference, it's because of how I cut the beet. This 10, I'll see a 0.1, a 0.217, a 0.204. So the ethanol and propanol isn't following the trend I would expect. Again, probably because of discrepancies in how I cut the beet. The 20%, 0.8, 0.18, 0.2, very close, and then 1.25. The 30%, 0.19, 0.41, and then 1.85, and the 40%, 0.33, 0.93, and 1.9. So you can see clearly the propanol has the most dramatic impact on the membrane. So now you can go back to the lab itself, and you can start looking at the guiding questions. You can look at the data. I'm going to put my own data. There's data that was on there already for the 3, but I'm going to put my own data up because it's accurate, and you'll be able to use that to help you answer these questions as you're going to that overall lab. So there is an overall lab in the course. Let me just open this up so we're all looking at the same thing. So when I come back here to calendar, and I go to complete the osmosis diffusion lab. So this will be due on Monday before class starts if you're doing this. Here's the fusion osmosis lab, the alcohol and this. So this here, these are the overall instructions. So this is including all the instructions for all the labs I demonstrated. There was one lab I didn't demonstrate, I really wanted to do, but again, there's only so much time, we can only do so many things. So you're welcome. So here it says, hypothesis for part 1, that's the one we did in class. Hypothesis for demo lab, so when the dialysis bag was filled with 100% syrup and submerged in the beaker of distilled water, will its mass increase or decrease? I'm actually going to change that, and I'm going to change that to reflect the eggs or the U-tube, so that way you can incorporate it a little bit better. And then you have the observations, but it's the red, that's what's going in your lab notebook. That's what I'm actually assessing. This analysis, discussion questions, the challenge question, and the conclusion, right? If you want to include those other things, you're more than welcome to, but it's the red that I'm focusing on for this particular lab. So if you want that feedback, it's in by Monday. Someone asked the question, if you don't get feedback, can you still submit it as a lab notebook lab? Absolutely, you just don't have that feedback, okay? All right, so I will end this here. That's the awkward ending. I think I'll edit that out.